Good morning, dear students. Today we are starting our class with another type of cell division, that is meiosis, or otherwise it is a reduction division. In the case of meiosis, from a diploid parent cell, a haploid, not single, four or like that, haploid cells are daughter cells are forming. And this type of cell division, meiotic cell division, occurs in sexually reproducing organisms. Sexually reproducing means in such organisms, zygote is forming by the fusion of egg and sperm. These are gametes, and their fusion forms the zygote. In such type of cases, if the parent cell, suppose the egg is 2n, that is diploid, and the sperm also diploid, then what result? The resulting zygote will be 4n or like that. 4n. So it may lead to abnormal condition, abnormalities. So we need to avoid that. Because of that reason, during the formation of gamete, reduction division is a must in the case of sexually reproducing organism. Suppose this is the parent cell, it is 2n, and it undergoes that meiosis. Meiosis, and by that way, what happens? The genetic content is reduced and become haploid, and the resulted zygote will be 2n, just like the parent. So in the case of sexually reproducing organisms, meiosis is a must to maintain the same chromosome number in the next generation also. Clear? We are going to see about different types of meiosis. Meiosis are of three different types: gametic or terminal meiosis, zygotic or initial meiosis, sporic or intermediate meiosis. First is gametic. Gametic, as the name indicates, in such type, the meiosis takes place at the time of formation of gametes. The formation of gametes, that time that meiosis is happening to reduce the genetic content to maintain the chromosome number in the next generation like that of the parent. Just as I described to you just now. Okay, such type of meiosis exists in which type of organisms? Multicellular animals, protozoa, and in few lower plants. All these ones are having this type of meiosis. In all those things, all such organisms, meiosis happens during the time of gamete formation. Next is zygotic or initial meiosis. That means the development of new organisms starts from the psychotic stage onwards, and that zygote is diploid. Then it undergoes meiosis to reduce its genetic content. I will show that. Suppose the formed zygote is 2n, and then it undergoes meiosis. And as a result, it later changed into haploid. Changed into haploid. That means such type of organisms that is having the zygotic or initial meiosis, the parent cell is haploid. That's why from such parent cell, fusion of the two counterparts results is diploid zygote, and for being the same as that of the parent cell, it is a must that it need to undergo meiosis to be in haploid state. So that is the zygotic or initial meiosis, and that type of meiosis exists in few algae, some protozoa and some fungi. Okay, clear. Next, the last type, that is sporic or intermediate meiosis. Intermediate, not that means that meiosis is not happening either in the beginning or in the final stage. Instead of that, it happens in some intermediate stage. It occurs in all higher plants. What is happening there? There, first male gamete and female gamete fuse to form, and show like that. Male gamete and female gamete, they fuse to form a diploid zygote, and this diploid zygote then develops to a diploid sporophyte. It is diploid, and to this sporophyte it undergoes meiosis. And for haploid spores. Haploid spores. And this haploid spores then germinate to haploid gametophores. Haploid gametophores. And this undergoes mitosis to form haploid gametes. So here that meiosis occurs in the intermediate stage, not in the beginning, not in the end of its life cycle. So that's why sporic or intermediate meiosis. It occurs in all higher plants. Okay. Then what is the main difference between mitosis and meiosis? In the case of mitosis, we have learned that it have only one series of successive division. One successive trial of division only present in the case of mitosis. But in the case of meiosis, two successive stages of divisions exist. That is like meiosis 1 and meiosis 2. But in the case of mitosis, we didn't see such type of two successive stages. In that, we learned only prophase, metaphase, telophase, and anaphase. But coming to meiosis, two successive divisions are present. Meiosis 1 and meiosis 2. That is one main difference. So many other difference also exist. One important difference is that one. Then pre-meiotic interface and interkinesis. These are two interfaces that present in meiosis. Pre-meiotic interface, as the name indicates, it is the interface period just before to the first meiotic division. Before the first meiotic division, there will be one interface. That interface is pre-meiotic interface. And in between the first meiotic and second meiotic, there will be another interface, that is interkinesis. Okay. Pre-meiotic interface. What is pre-meiotic interface? It is the preparatory phase before the first meiosis. What are the changes that is happening during that phase? The important process are the cell completes its DNA replication, and all the required RNAs are synthesizing its required proteins, carbohydrates, all those things are synthesizing during that time, and centrosome duplication. We have learned during the mitosis class itself, a duplication of centrosome is very necessary to begin the development of mitotic spindle. So that also finished during this pre-meiotic interface, and microtubules also started to extend from the centrosomes. These are the main changes that happens during the pre-meiotic interface. Next, we are moving to meiosis 1. or the first successive division in meiosis. It consists of four stages. Four stages. Name of those stages are the same that we have learned in mitosis. They are prophase, metaphase, anaphase, telophase. Only difference is that along with all, we are adding a one. Prophase 1, metaphase 1, anaphase 1, and telophase 1. Okay, then we can move to Prophase 1. What is Prophase 1? What all things are happening during that time? It is most elaborate and complex. In meiosis, this stage is the most complex stage. It consists of, it again contains so many sub-stages. 5 sub-stages are there. For memorizing each stages in the order, I'm giving you a code that is Lepsy square. Uh, so with that, you can easily memorize all the 5 substages of prophase in the order. Lepsy means leptotin, Z means zygotin, packaging, then di is 2 times. So diplotin and diakinesis. These are the different 5 stages that present in the prophase 1 of meiosis. Then we can go and see what is leptotin, what all changes are happening during leptotin. During leptotin, the main change happened is the condensation. The condensation of the chromosome begins during the leptotin stage. And along with that, an axial filament also start to form. What is axial filament? I will show that. Axial filament, in this picture we can see that it is a proteinaceous filament. It is forming along the sister chromatids. So these are the two main things that is happening during leptotin. So leptotin is over. Next is the zygotin stage, and this is very important stage, as during this stage so many important processes are happening. Important process of meiosis one is happening during this stage, and one such important event process is the synapses, occurrence of synapses. Synapses forms during the zygotin stage. Then what is synapses? Synapses is the pairing of homologous chromosome. Homologous chromosomes. What is this homologous chromosome? We know that we take a single human cell, it will contain 23 pairs of chromosomes. That means total 46 chromosomes. Um, that also indicates that the chromosomes exist in pairs. Pairs means if we take the case of first chromosome in a cell, uh, there will present two first chromosome, and one among this chromosome comes from mother and the other comes from the father. These chromosomes are called homologous chromosome. So in this zygotin stage, these homologous chromosomes that present in a cell are brought together. They come and align each other to form a set. That process is called the synapses. And there are 3 methods of that pairing. They include procentric pairing. Procentric pairing means pairing happens from the centromere towards the telomere in this direction. I will show that. Here need to show that. From the centromere towards the telomere. In this direction, that pairing starts. Such type of pairing is called the procentric pairing. And then pro-terminal pairing means what? Pro-terminal means the pairing starts from telomere towards the centromere. That is pro-terminal pairing. What is intermediate pairing? Intermediate pairing means that pairing happen simultaneously along the length at a same time. That is intermediate pairing. And after this pairing, we are calling each set as a bivalent or a tetrad. We are calling it as a bivalent or tetrad. Why it is a bivalent? Because each set, each synapses, it contains two chromosomes, and also each synapsed chromosome contain four chromatids. One, two, three, four. So it is a tetrad. Okay. Then another important thing that is forming during the zygote is the synaptonemal complex formation. Synaptonemal complex formation. Before that, we need to understand what is synaptonemal complex, what are its parts, all those things we need to understand. So I'm just going to show this is its structure. It is a tripartite structure. Tripartite means it consists of three parts. Tripartite structure with the two lateral element and a central element. Three parts are there for that synaptonemal complex. That synaptonemal complex help to hold the homologous chromosome together to undergo recombination. Once more I need to show, and along the side of the lateral element that chromatids are attaching with the synaptonemal complex. So that's what another important thing that is forming during the zygote. This is the structure helping to maintain the synapses and to support recombination. What is recombination? You will understand that process later in the section itself. Anyway, I just want to mention you simply. If we consider some daughters in them we can find a mixture of characters from mother and father. She neither looks exactly that of mother nor like that of her father. Instead, she is having a mixture of characters from both of them. For example, she is having a curly hair like that of her mother, and of having a fair complexion like that of her father. So that mixing up happened because of recombination, exchange of parts of the homologous chromosomes between each other. So for that purpose, that homologous chromosome come and form the synapses with the help of the synaptonemal complex. So this stage is very, very important. Its appearance also I already explained to you. It is a tripartite ladder-like structure. Three parallel axes are present, one central axis, two lateral axes, and one more point is there, transverse fibers. I missed that point. Transverse fibers, we can see that transverse fibers, it connects the two lateral elements together. That is the role of the transverse filament. Then to that lateral axis, chromatids are attaching. And from where that lateral axis emerged from the axial filament that formed during the leptotic period. Okay. And then one more thing I am going to tell you. How can we easily memorize that synapses is happening during zygote? One trick is there. Look at the first letter of stage, Z. This Z is somewhat similar to the first letter of synapses. By keeping that similarity in mind, we can easily memorize that synapses happens during the zygote. And that synaptonemal complex also, the first letter is S. Both that important process are happening during zygote. Okay. Next, we are going to see the significance of the synaptonemal complex. It gives a structural framework for recombination. I already told you, and it helps in synapses. And one more thing we can see in the synaptonemal complex, that is a recombination nodule. I'll show you. This is the recombination nodule. What is that? It is a multi-protein complex it contains so many proteins. And what is its significance? It helps in the, it is an indicator of the site where the recombination is going to happen. And such nodules we can see at regular intervals along the synaptonemal complex. At regular intervals. Okay, that is recombination nodule. Next stage is the packaging stage. It is the longest stage. We can remember that it is the longest stage. For packing anything before going to journey, we need so much time. So for packing, long time. So in that way, we can memorize. Packaging is the longest stage in meiosis one. And as a cell initiated the packaging stage, it indicates that synapses is over. Everything well set to have a recombination. In this stage, the important process that is taking place is crossing over. I will explain to you. Before that, we need to understand what are the changes that is happening during packaging. Uh, the homologous chromosomes are still held by synaptonemal complex, and also the chromosomes, its condensation is still continuing. So because of that, the chromosomes appear more packed during the packaging stage. Okay, and by the late of packaging, that homologous chromosome that form the synapses with the help of synaptonemal complex. Its non-sister chromatid undergo crossing over. We'll show that. These are the synapsed homologous chromosomes. After the completion of the synapses, the non-sister chromatids of the homologous chromosome. What is non-sister chromatids and sister chromatids? I want to make it sure that you know what is sister chromatids and non-sister. Look in this picture. Here, this chromatid and these chromatids are sister chromatids. This one and this one are sister chromatids. Okay, and if we consider this one and this one, they are non-sister chromatids. And those non-sister chromatids are undergoing crossing over during the packaging stage. And during the packaging stage, that crossing over is the important process. That also happen during the late packaging stage. Okay. That is, the crossing over is happening during the late packaging stage. These are the important changes happening during this stage. Next is diplotin stage. That stage is characterized by intense metabolic activity. And another significant process that happens is the formation of chiasmeter. What is chiasmeter? I will explain. Uh, During that packaging stage, I told you that crossing over happened during the packaging stage. And what is crossing over? That also I explained to you that this is the crossing over. Okay, and during the packaging stage, the two chromosomes are seen somewhat attached each other because of that synapses. And during this diplotin stage, what is happening is that these two chromosomes separate each other except at the point of crossing over. Then that point where the two chromosomes meet each other, touch each other. That point is called chiasmeter. Is it clear? Okay, I will show its image. This is a picture of chiasmeter. All the other regions start to separate each other, and only those portions where the crossing over happened, that region only stay together. Okay, that region is called a chiasmeter. Um, The chiasmata is of two types, terminal chiasmata and interstitial chiasmata. Terminal chiasmata means it is formed at the end of the chromosomes. Here, maybe here. And interstitial chiasmata means it may form some point along the chromosome in some other place other than the terminal portion. That is interstitial chiasmata. And Lamprecht chromosome is a particular appearance of the chromosome that we can see in the diploid stage. We have learned about this appearance of chromosome in the nucleus chapter in detail, so I am not going to explain it again. Uh, I only want to just brush up. What is Lamprecht? Lamprecht chromosome has a peculiar appearance in which to support the very necessary transcription process, the chromatin just loop out from the axis to give a brush-like appearance. And that form of chromosome, the chromosomes appear in that form is called the Lamprecht chromosome, and that appearance the chromosome show during which stage? Diploid stage. Then we need to understand what is uh, terminalization. Terminalization means crossing over is over and need to separate the two chromosomes. So for that, the chiasmata, I will draw and show you. This is the chiasmata, and for terminating or to bring about an end for that crossing over, the chiasmata need to move further towards the tip, and the point that previously remains together again gets separated. That is terminalization. After the first chiasmata gets separated, if some other chiasmata are present in some other points, that also move to the terminal point and get separated. And by the end of this terminalization, all the chiasmatas that present in the chromosome get separated. Okay, clear. That is diploiding, and these are the process and changes happen during the diploiding stage. Next stage is diakinesis. Diakinesis is the final stage of prophase 1 of meiosis 1. And what are the changes? It is almost similar to those changes that happen during the end of prophase that we have seen in the mitosis. Let's start. What are the changes? The chromosomes started to attach to the meiotic spindle. Here it is not mitotic, it is meiotic spindle. And the chromosome that is decondensed to some extent during the diploiding stage again recondensed during the diakinesis. Terminalization is complete, and nucleolus disappears. Nuclear envelope breakdown, a chromosome starts to move to the metaphase plate. Metaphase plate, everything I already explained to you. And meiotic spindle is formation has begun from the centrosome. All these changes we already have seen during the end of prophase in the case of mitosis also. And one more important thing we need to know, that is the arrest of meiosis. That means the meiosis, after reaching this stage, it is arrested in the case of female unborn babies. And when will it resume the further process? That happen only when that girl baby attains maturity or puberty. After she attains maturity, then each sex cell before ovulation time, each sex cells resume the further division process at the rate of 1 oocyte per month. Okay, that's very important. Next, we are going to see about the metaphase 1 of first meiotic division. What are the changes happening during this stage? Just similar to the metaphase stage of mitosis, here also that homologous chromosomes. In mitosis, there is no such homologous chromosome type arrangement. The difference is here. Here, the homologous chromosome are arranging in towards the metaphase plate, and that meiotic spindles are completely formed, and they come and attach with the kinetochore of chromosomes. And one more thing we can notice here. Here this blue chromosome got some portion of that red chromosome. That means it finished recombination. Recombination is over. That means, or in other words, maternal chromosome got some portion of paternal chromosome. Recombination is over. And then we need to count the number of chromosomes during this stage. 1, 2, 3, 4, 5, 6. 6 chromosomes we can find inside this cell. That is diploid condition 2n. Okay. These are the changes that happen during the metaphase 1. Next, we are moving to Anaphase One. Anaphase One, we know what's the basic process is that the chromosomes start to move towards the pole, opposite pole. The same thing is happening here also. Homologous here, homologous chromosomes that are previously remained together start to separate each other towards the poles. Okay. And these chromosomes, we can see that crossing over very clearly, and they are moving towards the poles. Okay, and because of that movement, after finishing that movement, we can clearly understand that each newly forming cell will get a haploid set of chromosomes. Okay, that we can see in the next slide. And one more thing we need to understand here, that segregation of the chromosome towards the two poles, it is random. It is not determinate which chromosomes are moving, which whether paternal or maternal is not able to determine it in that way. So that segregation of the homologous chromosome towards the poles are random. That also we need to understand. Okay. The telophase 1. Telophase 1 is a very brief period. It is the final stage of first meiotic division. And during this stage, in mitosis, after the end of telophase, the chromosomes again undergoes decondensation. But here, the chromosomes is not undergoing full decondensation during the telophase stage. And that separated homologous chromosome, they form a cluster at each pole. They form a cluster at each pole, and the nuclear membrane is reforming, but nucleoli is not reappearing at this stage. Then cytokinesis start, and this is in the formation of two independent cells, and the formed cells are haploid with respect to the chromosome number, but it is diploid with respect to the DNA content, right? The two cells are haploid with respect to the chromosome when compared with the parent cell. But as we look to its DNA content, it is still diploid. That also we need to reduce. Okay. Okay. Next, we are moving to the next interface that is between the first meiotic division and the second meiotic division. Is interkinesis. This is a stage between the two meiotic division. What are the features? We can notice. During this interface, distinct from the other interface, that is the pre-meiotic interface, here not much synthetic activities is taking place. Okay, and the cells that is in the interkinesis state in the case of animals, it is called secondary spermatocytes. It's called secondary spermatocytes, and if it is a female, it is called secondary oocyte. Okay. The point, one point again I am repeating, the cells are with a haploid chromosome number and with a diploid amount of DNA. Centrioles and astes again got duplicated. Okay, by here we finish with understanding the first meiotic division. Okay, clear? Next, we are moving to the second meiotic division. It resembles exactly like the mitotic division itself. Like that, it also possesses four stages: prophase, metaphase, anaphase, and telophase. As it is second meiotic division, these stages are called prophase 2, metaphase 2, anaphase 2, and telophase 2. Let me see what is happening in each phases. Prophase 2. Similar to the prophase of mitosis, here also the changes are somewhat similar. Nuclear envelope is breaking down, the mitotic spindle is start to form, and the chromosomes start to migrate towards the metaphase plate. These are the same process that happened during the that mitotic prophase. The same thing are happening here also. This is the prophase 2. Okay, that centrosome doubled, that already doubled during the interkinesis itself. Next, metaphase 2. By the metaphase 2, the chromosomes are completely correctly aligned in the metaphase plate, and it is attached with the spindle tubules, spindle fibers very correctly at its kinetochore. Then one more important thing is here, that's what? To proceed further to the next stage, that is the anaphase 2, that cell means that egg need to get fertilized by the sperm. Only then it can proceed to the next stage, that is the anaphase 2. So we can tell that the metaphase 2 is another stage that cell is having another stage of arrest. It is waiting to get fertilized. Okay. After it get fertilized, then it proceed to the next stage, that is the anaphase 2. During anaphase state, the chromatids of the chromosomes split up and they start moving to the opposite pole. That is happening here. We already have learned that in the former slide that at the end of first meiotic division, even if the formed two cells are haploid with respect to the chromosome number, still it is diploid with respect to the DNA content. And here by the anaphase 2, that DNA content also is reducing as that chromatids get split up and moves to opposite pole. Okay, clear? So the next phase is telophase 2, and the separated chromatids, they form groups and get enclosed inside the newly formed nuclear envelope. And after that, these chromatids undergo decondensation. By the end of this telophase 2, what resulted? 4 haploid nuclei. And after the formation of 4 haploid nuclei, the cell undergoes cytokinesis. Cytokinesis, as a result of that, it forms 4 haploid daughter cells. As the cytokinase also finish, that results 4 haploid daughter cells. Okay, then I am going to show you a generalized picture of this meiosis. And in the meiosis, we already have seen two arrest stages, which are the first arrest happens during diakinesis, that is during the end of the prophase first meiotic division, and it resumed after when the girl baby got mature, and the second arrest happens at the metaphase 2, and that from that arrest the cell resume the further division only after it gets fertilized by the sperm. Those two stages are the specific arrest period in the meiosis. And here I am showing a picture of oogenesis, how the egg is forming. Mm, this first one is a diploid cell. From that diploid cell, the primary oocyte is forming. It is diploid, and this from this cell, we are actually starting the meiosis. First meiosis is happening here, and we have learned that at diakinesis, the cell enters in an arrest period. Okay, so that everything happens here. Primary oocyte diploid that is present inside the body of unborn girl baby, and as she took birth and she got mature uh, at the rate of one primary oocyte per month, it resumed the further process of division. And as this single primary oocyte divide, it results two cells. And that first reduction, because of the first reduction division, the resulted daughter cell is having a half number of chromosomes when compared to the parent cell. But with respect to DNA content, it is still diploid. And that stage, that formed two cells, they have some specific name. One cell is called secondary oocyte, and the other one is called first polar body. Okay, and after forming, after reaching this stage, it again undergoes second meiotic division to reduce the DNA content also. And as this cell divide, another round of it undergoes another round of division. Again, two cells are forming from this, and as it undergoes another round of division, again two cells. A total of four cells are forming at the end of meiosis. Okay, and as this polar body undergoes division, the formed daughter cells are also polar bodies. And as the secondary oocyte undergoes second meiosis, the one cell that is formed out of the secondary oocyte is the egg, and the other one is called the second polar body. And this polar body will not sustain, it only dies off. Then what is the significance of these polar bodies? That these polar bodies are formed only to eliminate one half of the diploid chromosome set produced by the meiotic division in the egg to leave a haploid cell, that is the mature egg. Only then, without the formation of polar body, it is difficult to form a single haploid mature egg. Okay, is it clear? That's about the meiosis. Next, we are going to understand about the significance of meiosis. First significance is in sexual reproduction. In sexual reproduction, because of meiosis, four haploid daughter cells are forming, and the fusion of two such haploid cells, that is egg and sperm, result in the formation of a diploid zygote. By that way, it helps to maintain that diploid state of that organism. In that way, it is significant. And to maintain genetic diversity, meiosis play a significant role. In what all way it play that significant role, that I am going to tell you. Meiosis helps the reshuffling of genes. And which all way that reshuffling occurs? Suppose during that reduction division in meiosis, only one of the allele of a gene is coming to one gamete. We know that one gene is represented by two alleles. And if these two alleles are present in the next generation, it may give the same phenotype that present in the first generation. Okay, and because of meiosis, only one of this allele is entering the gamete, and it has the chance to pair with another allele and to form another type of combination, another type of gene, and can give a different phenotype with different phenotype with regard to the parent type. So in that way, it helps to maintain the genetic diversity. There's another method also, another process also happening during meiosis, that is the independent assortment of the maternal and paternal chromosome during Metaphase One and the Anaphase One. That also play an important role in maintaining the genetic diversity. That assortment, that movement of the chromosome towards the pole, it is random. So by that way also genetic diversity can maintain. Another method, another process that happens in the meiosis is recombination. Recombination also is an important process that helps to maintain the genetic diversity. And also the union or fusion of an egg, with which sperm. Because so many sperms are approaching the egg to fertilize it. Only one sperm is getting that chance, and that chance is random. Uh, Nobody can determine that. So that by that way also, the meiosis help to maintain the genetic diversity. For maintaining the chromosome number, we already understand that because of that reduction division that happened in meiosis, the cell can that diploid condition of the organism is possible to maintain. So these are the significance of the meiosis. With this, we finish the chapter cell cycle and today's section. Thank you.
I'm an icon, man. And my girl about a 10. I should fuck her in the Benz.